Good morning all students. Today I am going to discuss with the lactic acid bacteria. Okay, as the name itself says, the bacteria which produces lactic acid are generally termed as lactic acid bacteria. Okay, and in this conversion, one of the common pathway is like through the glycolysis, and from pyruvate lactic acid will be formed. Okay, these are some of the common pathways. So lactic acid bacteria are, most of them are gram positive non-sporulating bacteria that produces lactic acid as a measure or salt fermentation product from fermentation of sugars. And there are two basically fermentative patterns are fermentation patterns are being observed, of which one is homofermentative and the another one is heterofermentative. Okay, in the case of homofermentative, a single product, okay, which is a lactic acid, as we have studied lactic acid bacteria. So the word single, okay, so word homofermentative is used. Another one which is basically an heterofermentative, which yields, in addition to the lactate, okay, ethanol and carbon dioxide will be produced. Ethanol and carbon dioxide will be produced. Now moving forward with the some of the examples also. Okay, moving forward with the some of the examples. You can write down if you want. Okay, I will be repeating name 2 times for your convenience. Okay, so the group basically is streptococci, lactococcus lactis, lactococcus lactis. Next one, streptococcus salivarius. Next one, streptococcus salivarius. Next one, streptococcus pyogenes. Next group, lactobacilli. Okay, these are basically homofermentative. Okay, homofermentative type of lactic acid bacteria. Next one, in the lactobacilli group, lactobacillus delbrueckii, lactobacillus delbrueckii. Next one, lactobacillus helveticus, lactobacillus helveticus. Next one, lactobacillus acidophilus. Next one, lactobacillus salivarius. Next one, lactobacillus casei. Now, in the case of heterofermentative, okay, leuconostoc lactis, leuconostoc lactis. Next one, leuconostoc mesenterioids, leuconostoc mesenterioids. Next one, lactobacillus bifermentans, lactobacillus bifermentans. Next one, lactobacillus brevis. Next one, lactobacillus fermentum. Next one, lactobacillus candeleri. Okay, so these are some of the examples of hetero and homofermentative bacteria. Okay, now moving with the process. Okay, now moving with the process of homo and heterofermentative bacteria. Okay, as I told you, in the case of homofermentative, okay, the main product will be lactate. Okay, but there are also products, but main product almost you can say around 85 to 90% product will be lactate, but not all the 100%. Okay, and these bacteria, as you know, are basically found in the case of milk or even plants also, and in some case they are also found in the intestinal tract of the human beings and animals. Okay, so because in some cases due to the overproduction of lactic acid, In some cases, this helps for the various fermentation industries in the production of, as you all know, you have studied in your second year of semester 4. Okay. So, in the case of homofermentative lactobacilli, almost produce, as I told you, 80 to 85 to 90% of the lactate, okay, in which they metabolize glucose, via fructose biphosphate pathway, or more specifically glycolysis, okay, okay, and finally in this case pyruvate will be converted into lactate, okay, pyruvate will be converted into a lactate, okay. This is how a homofermentative pathway works, okay. In exam, okay, you can simply write the, in the case of homofermentative pathway, okay, you can write down whole glycolysis and step from pyruvate to the lactate for your exam, okay, for your exam. And as you know, okay, the you must know the conditions, that is, does it need aerobic conditions, does it need anaerobic conditions, we have already studied in our previous lectures, okay. And in the case of now heterofermentative, okay, in the case of the heterofermentative lactate fermentation, as the bacteria lack imported enzymes, okay, some of the enzymes, and it may lead to two different pathways, okay, two different pathways, okay. So in this case, two different pathways are involved for the production of lactic acid, but along with lactic acid, other products such as ethanol as well as carbon dioxide will also be produced, okay. Ethanol and carbon dioxide will also be produced at the end of the process, okay. So starting with the process, that is glucose, okay, and it will be converted into glucose 6 phosphate, and further it will be transferred to the 6-phosphogluconic acid. As you know, this one is a Antonov-Dudroff pathway, okay. Now which enters Antonov-Dudroff pathway, and ribulose 5-phosphate is formed, xylulose 5-phosphate, etc., and acetyl phosphate, which will be converted into acetaldehyde, and finally ethanol, okay. If you remember both the pathways specifically, it will be easy for you. So in this case, at the end, pyruvate, okay, pyruvate will be converted into lactate, and some of them, okay, from some of them, acetyl phosphate will be converted into ethanol, okay, as well as carbon dioxide. So in such cases, two different main products will be formed, okay. And some of the heterofermentative bacteria also convert acetyl phosphate either partially or completely to the acetate, okay, and transferring the energy rich phosphate bond to the ADP, and thus gaining utilizable energy as ATP. So in this case, excess of hydrogen is transferred to the glucose, okay, to give to the mannitol also, and the glyceraldehyde phosphate will be converted, okay, into the lactate via pyruvate, okay. One such example is of leuconostoc mesenterioids, okay, which ferments ribose to the lactate and acetate, okay. Now in the case of let's say lactobacillus plantarum, okay, in the case of lactobacillus plantarum, okay, which metabolizes glucose homofermentatively but splits pentoses by the enzyme photoketolase to the lactate and acetate, Okay, so it depends on the type of bacteria. Okay, in the especially in the case of heterofermentative, there are various mechanisms. Okay, the quantity of the ethanol and lactate may vary according to the different type of bacteria. Okay, and the enzymes which are being present in the system. So some of you may have question why two products are needed. Okay, it is up to us, it is up to human beings to need, they need a particular product, but in the case of bacteria they have their own mechanism which they have to work on. Okay, the bacterial metabolism works on the presence of the enzymes which are present in the bacteria. So of the some of the bacteria may not possess some different types of enzymes. Okay, due to which they undergo heterotrophic, okay, heterotrophic fermentation. Okay, this is how a heterofermentative fermentation works. Okay, so you can have an idea which two or three enzymes are missing in this pathway. Okay, they are the enzymes of glycolysis. I am not going to name it. Okay, you need to find out those enzymes. Okay, now some of the other examples, hetero and homofermentative. Okay, I'm just going to read out the terms or name of the bacteria. Okay, Lactobacillus lactis, Lactobacillus delbrueck, Lactobacillus bulgarius, Sporolactobacillus inu, Linus, Enterococcus facialis, Lactococcus chemoris, and Bifidobacterium bifidum. Okay, Bifidobacterium bifidum. From Lactobacillus, Sporolactobacillus, Enterococcus, they undergo homofermentative, whereas Lactococcus and Bifidobacterium undergo heterofermentative mode of lactic acid fermentation. Okay, they undergo heterofermentative lactic acid fermentation. Okay, so how biosynthesis occurs in the case of lactic acid bacteria. Okay, how biosynthesis occurs. Okay, it requires amino acids, vitamins, nucleic acid bases, and other substances as growth factors because they cannot synthesize. Them. So the pyruvate is oxidized to acetyl CoA also depending on the type of strain. Okay, and it may vary from bacteria to bacteria. Okay, it may vary from bacteria to bacteria. Okay, it may choose a different types of paths. Now the pyruvate dehydrogenase, as shown below, it's a multi-enzyme complex which oxidizes pyruvate to acetyl CoA in the more species of Lactococcus and Enterococcus, as in the case where they are aerobic bacteria. And the Enterococcus facialis and the Enterococcus facialis, Bifidobacterium bifidum, and Lactobacillus rely on the pyruvate format lyase, an enzyme found in the anaerobic metabolism in the facultative anaerobic enteric bacteria. Okay, so the mechanism depends on the type of bacteria. So here is one such small example given where pyruvate is being converted into acetyl CoA and formate. Okay, so as you all know, from acetyl CoA the pathway can move forward for the TCA cycle and also serve as an precursor molecules for the biosynthesis of various other biomolecules. Okay, now moving towards the end of our today's topic. The pyruvate oxidase and phosphotransacetylase converts pyruvate to the acetyl CoA in other lactic acid bacteria including Lactobacillus delbrueckii and Lactobacillus plantarum. Okay, and in this case pyruvate will be converted into acetyl phosphate. Pyruvate will be converted into acetyl phosphate in the presence of pyruvate oxidase, and this will be further converted into acetyl CoA. Okay, this will be further converted to acetyl CoA, and in other case, by the enzyme lactate dehydrogenase, pyruvate will be converted into lactate with the help of enzyme lactate dehydrogenase. Okay, so I am ending my today's lecture. Okay, please comment your name for the attendance and join the WebEx session of 8:40.